Good morning or good afternoon, depending on when you're watching this. First and foremost, thank you guys for your feedback on yesterday's e-learning. I'm recording this on the first day, obviously, but I've already gotten a lot of feedback this morning. And it seems like a lot of people like this format. A few people did have some suggestions, which I am looking into, so I will let you know what I come up with. But again, thank you guys for all the feedback. I really appreciate it. And I appreciate you guys being flexible during this time. I know this is a little bit strange. So for today, since yesterday we reviewed the process of DNA replication, because I know it had been a few weeks before that, today we're going to do a couple questions that go along with DNA replication. They're all, well, for the most part, they're all things we've talked about before. There's a few new things, but since I'm going through them with you together, I will give you the answers here. I'm going to go through these. There are 15, I believe, questions that we're going to do today, but there are more questions past that point. We'll come back to those questions later when we get to DNA transcription and translation. I put a slide on there that says end of day 1. You don't have to go past that. Just make sure as you're following along, I've posted these questions on a Google form as well on Google Classroom. They all just require short answers, and I have them on every slide here, just like 1 or 2 word answers for most of them. So make sure you're following along. You can even have that pulled up and have this playing in the background as you go through, or in a different tab. So as I go through these, just fill these out on the Google form, or you can go ahead and work ahead and fill them out on your own if you feel comfortable. So let's go ahead and get through this. So the first one should be, I want to say easy, but the word is very complicated. So DNA stands for deoxyribonucleic acid. We talked about this in class, but I don't expect a lot of you to remember that word because it's a lot of syllables. Not super important for your understanding, but it's just a good fact to know. And again, if you're like me and you understand why a word is the way that it is, it helps you understand what that thing is meant to be. So deoxyribo, meaning the sugar that's found in it, nucleic, meaning it's found in the nucleus, and acid, because it's an acid, which is a chemical property, which you'll talk more about when you get into chemistry. Question number 2, what is the function of DNA? Hopefully you know this at this point. The function of DNA is to control the formation of proteins in your body that determine who you are, what you can do, all of your physical traits, stuff like that. It determines everything about you, from the physical makeup of your body even to things like your personality and what proteins you can make and what you can digest and things like that. So everything about you is controlled by your DNA. It's the information, it's the instructions. I'm going to keep moving, but again, this is a video, so feel free to stop me and pause at any point. 3 parts of the nucleotide. We've gone over these several times, but I know it's been a while, so if you don't remember all of them, here they are again. If you remember, these are the 3 major parts, and if think of these like links in a chain, and if you put these back to back, you can make a DNA molecule. But each part has these 3 things in them. It has a phosphate group, usually indicated by a circle. That's not actually what it looks like, obviously, because it's a molecule, but for simplicity's sake, we'll just draw a circle for the phosphate group. A 5-sided sugar over here, and then you have the base. And again, these bases come in 4 varieties: A's, G's, T's, or C's, which we'll get onto in another question. So there's the base. There are the 3 parts there. Feel free to pause if you need to see these. Oh, I just talked about this. So 4 nitrogen bases found in DNA. So going back to these bases, again, this is the only part that can vary between DNA molecules. You've got A's, G's, T's, and C's. And if you remember Chargaff's rule, A's will always go to T's and vice versa, and G's will always go to C's. So if you have A's on one side of the DNA molecule, on the opposite side you'll have T's, and vice versa. And if you have G's on one side, you'll have C's on the other side. This is how the DNA knows where to put certain base pairs. It's not entirely random. DNA nucleotides are joined by covalent bonds between the sugar group and the phosphate group going down the side. So what this is saying, because this is worded a little bit confusingly, is in order to keep these molecules connected, you can see here each of these nucleotides. So here's the 3 components we just talked about, repeating over and over and again, and they're combined and they're connected at the sugar and the phosphate group like a ladder, so just going down the side there. If that doesn't make sense, then that's fine, just copy these down. Nitrogen bases in DNA are held together. We have not talked about this one, and it's not super important, but I thought I would include it just in case. I don't know if this will come up in chemistry, if Mrs. Snowballs talks about the different types of bonds at all. I would assume that she does, but I don't know if she talks about these types. But just in case, hydrogen bonds. All you need to know about hydrogen bonds right now is that they are some of the weakest bonds that you can form between molecules, which is how you can separate the DNA so easily. Most chemical bonds are fairly, at least compared to hydrogen bonds, they are stronger than a hydrogen bond. Hydrogen bonds are very weak in comparison, which is what allows them to be separated so easily, which is important for when you want to copy the DNA. Shape of the DNA molecule, like it is shown here, the most famous depiction of the DNA molecule. It's a twisted ladder called a double helix. We've talked about this before, and I'm assuming even before this class you've heard this term. It's basically a ladder that's twisted around itself, if you are confused about what that means, but I think everyone's heard that term before. Moving forward. The The double helix shape of DNA was first described in 1953. This was on our PowerPoint originally, and I will try to remember to upload that as well, but if I don't, you can always go back and look at it. Just a little bit of the history behind it. It was discovered by James Watson and Francis Crick. They were the first people to finalize the shape of it, but there were actually several people involved in the discovery of it. It's not like one day 2 people came along and they suddenly got it. This was years of work done by several people, most of whom did get credited for this discovery. In addition to these 2, it wasn't just these 2, it was a lot of people with a lot of work behind it. So process of copying DNA is called DNA replication. That's what we've been spending the last couple lessons on. This is what we were working on before break. If you remember this is depicting interphase, the first phase of mitosis. We won't be talking about mitosis again because we already did that, but it is related because this is the phase in mitosis where the DNA replicates. Briefly explain why DNA replication is necessary. So, here's a short answer if you don't want to hear me ramble. Long answer, for multicellular organisms that are made of more than one cell, obviously, if you're going to make more cells that are going to make up another organism, you need to have instructions for that cell. So for every new cell, it needs a set of instructions. And then even for single-celled organisms, even though they are single-cellular, they still need to reproduce at some point, and those offspring still need to have instructions for how to run their lives. So anytime a cell replicates, it needs to give a copy of DNA to whatever the new cell is going to be, because without it, the cell will not be able to function. This is another one we've not talked about, not super important for right now, especially since we're going to be restricted to distance learning for the rest of the year, but if you come back, we might talk about this in Bio 2, and probably not anatomy. I don't think this will come up in anatomy, but just in case. During DNA replication, the molecule unzips at what is called the origin of replication. If you remember, I said that DNA is copied in multiple different places all at the same time. If I told you to copy down a book, it'd take a lot longer to copy it from start to finish in one continuous piece. It'd be a lot easier to do it in chunks and to delegate parts of the book to different people, so that's what's going on here. You can start in various different parts along the molecule, and where these parts unzip, where that bubble forms, so where it's opening up, is called an origin of replication, and those can happen in various different places throughout the molecule. The result of DNA replication is 2 new daughter molecules. Same kind of concept when we talked about, again, mitosis. We talked about how they form daughter cells. For whatever reason, when we're talking about things like this in biology, they typically term the resulting molecule or the resulting cell as a daughter cell, like you would a child and parent. So these new molecules are called daughter molecules. I don't think that'll come up a whole lot, but just in case you hear that term, that's what that means, and I think you could piece it together even if we didn't talk about that. This will be really important moving forward because a good chunk of what we're going to be doing is related to this kind of question. Because even when we get on to transcription and translation, a lot of transcription and translation is also very similar to this as well. So if nothing else from today, if you are confused about this, please let me know because it's only going to get more complicated past this point, and then we're going to add some more rules. So list the bases that will base pair to this section of DNA replication. So if you remember, A's will go to T's and vice versa. C's will go to G's and vice versa. Bit of foreshadowing for next week when we get into transcription and translation. That rule will change, and I apologize for that because it's going to get confusing and it's going to make you probably mess some things up if I'm just being honest, but that's just because of the nature of it. But for now, we're going to keep it simple. A's to T's, T's to A's, G's to C's, C's to G's. So we have ATT, CAG, CAT, CCG. If you already know how this works, then this should be fairly simple. A's will go to T's, so there should be a T. T goes to A, so T, A, A. G for C, T, C, G, T, A, G, G, C. So I know that was a lot. Here it is. If you need to see it all written down, pause it if you need to. Hopefully that makes sense. Email me if you have any questions about that, but that should be fairly simple at this point. Again, please ask questions if you have any about this because we're only going to add more rules to this as we move forward. So if you don't get it now, it's just going to be even more confusing getting past this point. Okay, almost done. I think there's 1 or 2 more. Your DNA contains the instructions for making your genes, and these determine your traits, and this kind of goes back to what we do. Punnett squares, you can see down here, it talks about how genotype controls your phenotype. If you remember, genotypes are your genes, your alleles, your A's and B's, and whatever letter you choose to use. Big A's and little A's, dominant recessive traits, and that will determine what your phenotype is. So if you have a recessive trait, you will show attached earlobes or brown eyes or blue eyes or whatever it may be, depending on whatever the trait is. All right, and this is the last one, I believe. Yeah, there's the answer. So we'll wrap up on this. We haven't talked about this a whole lot yet, but this is good foreshadowing for next week, like I said, because we're going to have to get into the differences a little bit more when we get to transcription and translation, because transcription is going to incorporate RNA, which we have not talked about a whole lot yet. So if it doesn't make sense yet, keep this in the back of your mind, because this will be important for next week, and we can always come back to this if you need it. But DNA and RNA. Here are some major differences. These aren't all the differences, but these are the most important ones for today at least, but we'll talk about a couple other ones. DNA is double-stranded, whereas RNA is single-stranded. That just determines a little bit of its function, a little bit about how it works and how it reads. We won't get into specifics for right now because it's not super important for today's discussion. But anyway, another difference has to do with the name. So this one's DNA, this one's RNA. The D and the R come from their names. DNA is for deoxyribose. Ribose is a type of sugar. So this one has just regular ribose. This one has deoxyribose, which for simplicity's sake is a ribose without an oxygen on it. It got an oxygen taken off. It's deoxygenated, deoxy, which is where the names come from. Biggest thing, one that we've not talked about, well, we talked about it a little bit, but not gotten to a whole lot of detail on, are the bases. Almost all the bases are the same between DNA and RNA, but there is one major difference that is going to affect what we're going to look at when we do transcription. We've talked about how DNA has A's, T's, G's, and C's. RNA has A's, U's, G's, and C's. So these three are the same, A's, G's, and C's. However, T's get replaced with U's. I apologize for that, but it's not something that people can control. That's just biology. That's just how it works. That's just what the molecule is. So it's just going to have to take some getting used to. So next week, we're going to spend a lot of time trying to switch gears and flip back and forth between replication and transcription, because with replication, you're going to be using T's. With transcription, you're going to be using U's, and it's just a matter of flipping back and forth. Most of the time, flipping the letters is easy for a lot of people. What a lot of people mess up on is knowing when to use U's and when to use T's, and that's understandable because it's a very minor difference. So just keep that in mind. I'll talk about that more next week if that doesn't make sense. But either way, that should be all the questions for today. Hopefully I kept that short and concise. I know I tend to go on and on, so I'm going to try and do a better job of keeping these short, because I know you guys have other stuff to do throughout the day. So again, feel free to go back through this video at any point if you need anything, but I'm also posting this PowerPoint in addition to some other resources if you need them. Email me if you have any questions. And then tomorrow we are going to start on, we won't finish it because I think it would be a lot to do in one day, but we're going to start on a virtual activity that I would typically do in class. I'm going to introduce the topic tomorrow, and then I'll probably run through the actual simulation on Tuesday. That way we can kind of chunk it up. It's going to be talking about PCR, which is a lab procedure that I'll talk about tomorrow. But basically the gist of it is that it has to do with DNA replication and copying DNA in a lab setting. So why would we do that? What would we need it for? So on and so forth. So I'll get into that tomorrow. Let me know if you have any questions. So far so good. It seems like things are working out so far, but again, keep letting me know if you guys have suggestions or questions moving forward, and we'll keep chugging along as we keep moving. So I will see you guys tomorrow, and let me know if you have any questions.